BTEC Human Biology Unit 1, A3, the Cardiovascular System Summary. So first of all, looking at the composition of blood. So plasma is the liquid part of the blood, and it's made up of 90% water, plasma proteins, salts, amino acids, glucose, urea, and carbon dioxide. Red blood cells, also known as erythrocytes, their role is to transport oxygen, and they have a biconcave disc shape to create a large surface area to volume ratio, which means there is faster diffusion of oxygen. There is also no nucleus, which increases that surface area available to carry oxygen, and it contains hemoglobin, so it can carry more oxygen. There is also white blood cells, known as leukocytes. These are for defence and immunity, and they detect foreign material and destroy it. Then you've got platelets, also known as thrombocytes. These promote blood clotting, and the three main types of blood vessels are arteries, veins, and capillaries. So here is the general pattern of blood circulation in a mammal. So you can see it's got a double circulatory system. All of the parts shown in red are oxygenated. All the parts shown in blue are deoxygenated. So it's got a double circulatory system because it goes to the lungs, which is called the pulmonary circulation, goes to the lungs to pick up oxygen and then back to the heart. And then it gets repumped to send the oxygenated blood around the body and back to the heart. Anything to do with the liver has the word hepatic in it, so you've got hepatic artery and a hepatic vein. You've got the intestinal artery, and from the intestines to the liver, you've got the hepatic portal vein. Anything to do with the kidneys has the word renal in it, so you've got the renal artery takes blood to the kidneys. The renal vein takes blood away from the kidneys. So, the circulatory system of a mammal is a closed double circulatory system that has two circuits. So blood passes through the heart twice for each complete circulation of the body. So pulmonary circulation is where deoxygenated blood in the right side of the heart is pumped to the lungs, and oxygenated blood returns to the left side of the heart. Systemic circulation. Oxygenated blood in the left side of the heart is pumped to tissues or organs of the body, and then deoxygenated blood returns to the right side. This double circulatory system is important for mammals because it prevents the mixing of oxygenated and deoxygenated blood. So blood is pumped to the body and it is fully saturated with oxygen. This is an efficient way of delivering oxygen and glucose for respiration. Blood can be pumped at a higher pressure after being lowered from the lungs. Substances are taken to and removed from the body cells quicker and more efficiently. The coronary arteries are the arteries that deliver oxygenated blood to the cardiac muscle. Blood vessels entering and leaving the heart. So the aorta takes oxygenated blood from the heart to the respiring tissue. The vena cava takes deoxygenated blood from the respiring tissue to the heart. And there is also the pulmonary artery and pulmonary vein. Blood vessels entering and leaving the lungs. The pulmonary artery takes deoxygenated blood from the heart to the lungs. The pulmonary vein takes oxygenated blood from the lungs to the heart. Blood vessels entering and leaving the kidneys. Renal arteries take deoxygenated blood to the kidneys. Renal veins take deoxygenated blood to the vena cava from the kidneys. How the structure of the heart relates to its function. Atrioventricular valves prevent the backflow of blood from the ventricles to the atria. The semilunar valves prevent the backflow of blood from the arteries to the ventricles. The left side of the heart has a thicker muscular wall, so it generates a higher blood pressure. For oxygenated blood has to travel greater distance around the body. The right side of the heart has thinner muscular walls. This generates lower blood pressure for deoxygenated blood to travel a small distance to the lungs, where high pressure would damage the alveoli. The structure of arteries, arterioles and veins in relation to their function. So you can see that arteries are larger vessels. They branch into smaller arterioles, then into even smaller capillaries. They join back into venules and then back into one vein. Arteries carry blood from the heart to the rest of the body at high pressure. They have a thick, smooth muscle layer which contracts, pushing the blood along, and controls or maintains the blood flow or pressure. Has elastic tissue to stretch as ventricle contracts under high pressure, and recoil as ventricle relaxes under low pressure. Reduces pressure surges even though the blood pressure, and maintain a high pressure. Thick wall withstands high pressure and prevents artery bursting. Smooth endothelium reduces friction. There is a narrow lumen which increases and maintains a high blood pressure. Arterioles are divisions of the arteries to smaller vessels which can direct blood to different areas. Thicker muscle, larger than arteries, constrict to reduce blood flow by narrowing the lumen, dilates or relaxes to increase blood flow by enlarging the lumen, thinner elastic layer as lower pressure surges. Veins carry blood back. They have a wider lumen than arteries. There is very little elastic and muscle tissue. They have valves which prevent the backflow of blood. There is contraction of skeletal muscles which squeezes the veins which maintain blood flow. Structure of capillaries and importance of capillary beds at exchange surfaces. Capillaries allow the efficient exchange of gases and nutrients between blood and tissue fluid. The capillary wall is a thin layer, only one cell thick, of squamous endothelial cells. They have a short diffusion pathway for rapid diffusion. Capillary bed is made of a large network of branched capillaries which are all thin. This increases the surface area to volume ratio for rapid diffusion. They have a narrow lumen which reduces the flow rate, so more time for diffusion or exchange. Capillaries permeate the tissues and no cell is far away from the capillary. There is a short diffusion pathway. There are pores in the walls between the cells, and it allows substances to escape, for instance, white blood cells to deal with infections. So, myogenic stimulation of the heart and transmission of a subsequent wave of electrical activity. Roles of the SAN, AVN and Purkinje tissue in the bundle of His. So, cardiac muscle is myogenic, and it can contract or relax without receiving electrical impulses from nerves. The sinoatrial node, or SAN, acts as a pacemaker and sends regular waves of electrical activity across the atria, causing the atria to contract simultaneously. Non-conducting tissue between the atrial ventricles prevents impulse passing directly to ventricles, preventing the immediate contraction of ventricles. Waves of electrical activity reach the atrioventricular node, or AVN, which delays the impulse, allowing the atria to fully contract and empty before the ventricles contract. The AVN sends a wave of electrical activity down the bundle of His, conducting wave between ventricles to the apex where it branches into Purkinje tissue or Purkinje fibres, causing the ventricles to contract simultaneously from the base up. Roles and locations of chemoreceptors and pressure receptors, and the roles of autonomic nervous system and effectors in controlling the heart rate. Chemoreceptors and pressure receptors located in the aorta and carotid arteries. The medulla oblongata in the brain controls heart rate via the autonomic nervous system. There are two divisions: the sympathetic nerves, which increase heart rate, and the parasympathetic nerves, which decrease heart rate. When you exercise, or if your blood pressure is too low, baroreceptors detect a fall in blood pressure, and chemoreceptors detect blood rise in blood carbon dioxide. They send impulses to the medulla oblongata, which send more frequent impulses to the SAN along the sympathetic nervous system. So therefore there's more frequent waves of depolarization sent from the SAN and the AVN, so the heart rate increases. When you relax, or if your blood pressure is too high, baroreceptors detect a rise in blood pressure and chemoreceptors detect the fall in blood carbon dioxide. They send impulses to the medulla oblongata, which send more frequent impulses to the SAN along the parasympathetic nervous system. So less frequent waves of depolarization are sent from the SAN and AVN, so the heart rate decreases. Coronary heart disease. An atheroma is a fatty deposit within the wall of an artery, and a thrombosis is a blood clot. An embolism is the sudden blockage of a blood vessel by an embolus. Coronary heart disease, or CHD, is a narrowing of the small blood vessels that supply blood to the heart. A myocardial infarction is a heart attack. Coronary heart disease risk factors: family history, smoking, high blood pressure, high cholesterol, obesity, diabetes, high stress, physical inactivity. Symptoms of coronary heart disease: chest pain, pounding heart, heartbeat, myocardial infarction. Stroke: a sudden attack of weakness or paralysis that occurs when blood flow to an area of the brain is interrupted. Stroke symptoms: sudden weakness or numbness of the face, arm or leg, especially on one side of the body. Sudden dizziness, loss of balance or coordination, or trouble walking. Sudden confusion, trouble speaking or understanding. Sudden trouble seeing, sudden severe headache. Stroke risk factors: high blood pressure, heart disease, smoking, high red blood cell count, females during pregnancy. The formation of tissue fluid and its return to the circulatory system. Tissue fluid is the fluid surrounding cells or tissues. It provides respiring cells with water, oxygen, glucose and amino acids. It enables waste substances to move back into the blood, like urea, lactic acid and carbon dioxide. The formation of tissue fluid at or nearest the arterial end of the capillaries. There is higher blood or hydrostatic pressure inside the capillaries due to contraction of the left ventricle than the tissue fluid. Therefore, there's a net outward pressure. This forces fluid or water out of the capillaries. Large plasma proteins remain in the capillary. The return of tissue fluid to the circulatory system towards the venule end of the capillaries. Hydrostatic pressure reduces as fluid leaves the capillaries. An increase in concentration of plasma proteins lowers the water potential in the capillary below the water potential for tissue fluid. Water re-enters the capillaries from the tissue fluid by osmosis down a water potential gradient. Excess water is taken up by the lymph system and is returned to the circulatory system.